Hi everyone, this is Dr. Ashiq from Department of Zoology, Haji Kartara Haudia College, Uttamapalayam. I am here to throw some light on external morphology of fishes. So I've incorporated the details pertaining to the external morphological features present in a typical fish. So uh, the picture right in front of you is the typical fish. So what are all the features present in a typical fish? When I say typical fish, this means it is a hypothetical fish. Some of the features may or may not be present in different species of fishes. So that is why the term typical fish is mentioned here. So external morphology, as we all know, the phenotypical characters, the characters we see with our naked eye, the characters which define the organism by seeing it. So those are called the external morphology, right? So the fish here is a bony fish. It's a teleost fish. So when we take a fish for a study or a scientific examination, we will keep the fish like this. So that the features of the fish will be clearly visible in such a way that we could study it clearly and legibly. Okay, so it is divided into two parts, right? So it is based on the sides, right? The head side, the fish body, let us divide the fish body into two halves, right? Right in the, let us divide it, let us bisect the fish body into two. So the side of a fish which has the head region is called anterior side, and the opposite side which comprise of a tail is called as posterior side. The upper side is called dorsal side, and the belly, the bottom side, is called as ventral side, right? So after this, let us divide the body of the fish and we study the features present in each region. First, head region. Head. The head is comprised of a pair of eyes and a pair of nostrils and a mouth. Inside the mouth, there is one or there are few rows of teeth, right? Then, besides the mouth and eye, there is a lid. There is a lid which covers the gill slits. The gill slits are present inside that lid. The lid is called operculum. Operculum, which is mentioned here. The gill cover, right? So inside the gill cover, if we open the gill cover, we could able to see the gill slits. Gill slits. செவில் அப்படின்னு தமிழ்ல சொல்லுவோம். செவில். Right. So it's quite common when we buy a fish from the market or from a vendor. We used to check whether the fish is fresh or it's been caught some days ago. How do we check? We will check by examining the gill slits, the color of the gill slits and the freshness of the gill slits, right? So for that, we have to open that operculum, the lid. So the part which is the reddish filamentous part which is present inside the lid is called operculum, gill slits, right? Okay. Near to the right from the operculum, a line will starts and run towards the end of the caudal fin. That line is called lateral line. Lateral line. So this line extends from the distal edge of operculum to the end of caudal fin. To the caudal fin. Okay. This line also differ in different species. So either way, it is present on the lateral side and hence the name lateral line. Right. Then after this, in the body region, we can see the fins. Fins are two types based on its occurrence. Whether the fins are two in number or the fins are in pairs, it is called paired fins. And the other type where only one fin is present, right, which is unpaired fin. Okay, so we can see the large fin present on the dorsal side, which is called as dorsal fin. Dorsal fin. Then exactly opposite to the dorsal fin, a paired fin is present, which is called pelvic fin. Pelvic fin. And before pelvic fin, there is a fin called pectoral fin. Pectoral fin and pelvic fin, both are paired fins. On either side, you can see a fin. If we see the pectoral fin from the left side of the fish, there will be the same present on the right side as well. Same goes to pelvic fin also, but the distance between the pelvic fin is much more smaller or very less when compared to the distance between two pectoral fins, because pectoral fins are attached on the lateral sides, one on the right side and the other one on the left side, whereas pelvic fin are lie under the belly, which is closed, which is close to each other, which is close to each other. Okay, so these pectoral fin and pelvic fins are paired fins, right? Okay, then opposite to pelvic fin, we already saw there is a dorsal fin. In some species of the fishes, the dorsal fin is two in number. There are two dorsal fins you can see in several fishes, like mullets belongs to the family Mygilidae. Those fishes have two dorsal fins. In that case, the first one is named as first dorsal fin and the second one is named as second dorsal fin. Right. In this case, some of the fish species, instead of second dorsal fin, they have a small extended structure, which looks like a fin, but it is not actually a fin. It is called as adipose fin. Adipose fin. Why we are calling it? It is not a fin because each and every fins we see, like for example dorsal fin or pectoral fin or pelvic fin, all of those fins are comprised of rays. If we can see all the fins, there are so many rays running through the fins. All along the fins, we can see the number of rays running through it. It is like a skeleton for fins, right? So those rays cannot be seen in adipose fin. This adipose fin is only comprised of fat bodies, and hence the name adipose fin, right? Okay, exactly. Then next, opposite to the adipose fin, there is anal fin. Why the name anal? It is because it follows the anus. It is present after the anus. So it is mentioned in this picture as vent. Vent is nothing but anus. So after that excretory pore through which the fecal matter will comes out, right? The excreta will comes out through vent. So since the fin is presented post vent or post anus, it is called as anal fin. Anal fin. And then next to anal fin, there is a stem-like cylindrical structure, which doesn't have any fins. That particular region is called peduncle. Peduncle, the stem, which means the stem. Then peduncle is followed by tail fin. Tail fin is called as caudal fin. Right? These are the important features present in a typical fish. As I said earlier, this doesn't mean all the features are present in all the species of fish. Some species lack dorsal fin, some species lack caudal fin, some species may lack pectoral fins. So it depends on the species. And that's why I said it is a hypothetical fish. Let's move on to the next one. So this again is a typical fish. It shows the measurement of a fish, how to measure a fish. These measurements, you can see right from all the measurements start from the snout region. It starts from the uh, farthest end, and it starts from the beginning and end at the farthest end, which is the tail, right? So each and every measurements are given a name. For example, head length. Head length starts from the tip of the snout until the operculum. Until the operculum. So from snout to operculum is the head region. So the length of that particular region is called as head length. Let's take a total length. Total length is the complete length of the species, fish species. It starts from the snout and ends in the distal part of the tail, the farthest part of the tail. It is called total length of the fish. So likewise, all the area has been given a name. So why these measurements? These measurements play a vital role in species identification. To identify a fish species, these measurements are mandatory. These measurements are called morphometric measurements, right? Okay, these morphometric measurements are vital in identifying the species, whether and also this is very important to study the biology of that particular fish species. Biology, when I say biology, that includes age, growth, length and weight relationship of the fishes, etc. So even we could predict the stock assessment by or the population dynamics of that particular fishes can be studied using these morphometric measurements. Okay. As I said earlier, there are so many variations. Some fishes lack uh, any fins. Some fishes lack some characters. So those typical special fishes, we can say special fishes that are presented here, that we can see. On the top right is the sunfish. Sunfish, which is called as mola mola. Okay, if you see the sunfish, one cannot say that it is a fish, right? It doesn't even look like a fish. Okay, it only has the dorsal fin and the other one on the ventral side. Even a pectoral fin is present, right? There is no distinct caudal fin. There are no distinct other parts of the fish, like operculum is not there. We can hardly find even a scales on the body, right? So each and every fish species are distinct and different from each other. Okay, so all these fishes are modifications of different morphological characters, right? Down below is the ray fish. திருக்கை அப்படின்னு சொல்லுவோம் in Tamil. திருக்கை மீன் அப்படின்னு சொல்லுவோம். So, and on the bottom left is விலாங்கு மீன், eels, eels. Above the eel is suckerfish, sea lampreys, petromyzon. We used to say petromyzon, sea lampreys, or simply a suckerfish. It is an ectoparasite of many large active fishes like sharks and other fishes, right? So these are all the exceptions to the typical fish. And as I said, some of the fins are paired fins, and the other fins are unpaired fins. Let's take pectoral fins. Pectoral fins are like our front uh, limbs or forelimbs. Let's say we have a pair of hands. We have a pair of hands. So instead of, it functions in a similar way. It is homologous. That is why it is written as homologous to forelimbs. Similarly, pelvic fins, which functions as, or which is homologous to hind limbs or our hind legs, okay, which we use to run or walk, etc. So these two fins form the forelimbs and hind limbs of the fishes. Then the position of pelvic fins varies from species to species. We can see in these two fishes, the position of pelvic fins vary. In the first fish, it is in the abdominal region. Abdomen, what is an abdomen? It's a belly, right? In the belly region, in the pelvic fin room. Whereas in this fish, the fish in the bottom, which there the fins, pelvic fins are close to the thorax region, which is next to head. Okay, which is in between head and abdomen. So, and hence the name thoracic pelvics, right? So each and every character differs according to the fish species. Okay. So as I said earlier, not all the features are present in all the fishes. Let's take, for example, eels and sunfish, mola mola. On the right side is the sunfish. They don't have pelvic fin at all. They don't have pelvic fin at all. See in the picture, it has a dorsal fin, and opposite to that is an anal fin or ventral fin. There is no pelvic fin. In a similar way, sea lampreys or petromyzons or the suckerfish, even those fish doesn't have pelvic fins, and also it lacks pectoral fin as well. So they lack pectoral fin as well as pelvic fin. So in eels and sunfish mola mola, they lack only pelvic fins. Then fins. So as we already seen, the pelvic fins, pectoral fins. Number one is pectoral fin, which is highlighted in pale red color or pink color, we can say, right? Which is called, it is a paired fin. It is paired fin present one on both the sides. Present one on both the sides. Okay. Then next to pectoral fins are pelvic fins. These pelvic fins are also paired fins. Apart from these two fins, all the other fins are unpaired fins. Say for example, dorsal fin. Dorsal fin is unpaired fin. And anal fin again is an unpaired fin. Means that it occurs in only one in number. And also adipose fin and also the caudal fin. Okay, caudal fin, again, caudal fins are different for different species. As we can see in the first picture, though there are four types of caudal fins. It is named as heterocircle, which means both are different. If we take a typical fish, a caudal fin, we see there are two lobes. Two lobes will be there. Both the lobe, the upper lobe and the lower lobe will be more or less equal. They look almost the same, right? There is a symmetry. Symmetry will be there. If the symmetry will be there, then it is called homocircle. We can see these homocircle tails in so many of the fishes. Both the lobes are equal, the upper lobe and the lower lobe. There is a symmetry, but whereas the symmetry is biased towards any one side, in that case, based on that, it is number one, heterocircle. Heterocircle tail, it is asymmetrical. The upper lobe is somewhat higher in length when compared to the lower lobe. As we can see those type of tails in sharks. Sharks, we can see that. And also in sturgeon fish. Sturgeon fish also, it will be there. Heterocircle tail. Then the second type is diphycircle. Diphycircle. Both the upper lobe and lower lobe will be connected by the small intermediate lobe. We can see the three lobes, the upper lobe and lower lobe will be connected by a small lobe. And hence this type of tail is called diphycircle tail. This will be seen in coelacanth fishes. Coelacanth fishes, the primitive fishes. And the third type is protocircle. Protocircle tail is also symmetrical. And we can see the last vertebra will run all along the body. We can see the gray area will be running until the end of the tail fin. Whereas you can see all the other three types where the gray part will be ended in some ended before, way before the fin starts, unlike the protocircle. Right. Okay. Then most of the fishes will have homocircle tails. Homo means same, right? That means homo means same or equal in length. So the upper lobe and the lower lobe will be more or less equal in size. So then again, the homocircle tail is further divided into different types. Whether the tail end is rounded, when it is spherical in shape or semicircle, then it is called rounded tail. We can see from the shapes, rounded tail. And when it is deeply forked, you can see the two clear divisions in the tail region. Then it is called forked. Forked. And the other one is lunate. Lunate. When it is looking like crescent, lunar refers to moon, and hence when it looks like a crescent, then it is called lunate. And when it is square or like a straight, when we it's like a straight line or square shape, then it is called truncate tail. Similarly, there are several other types also, but these four types are quite common in most of the fishes. Then comes to scales. Scales. செதில்கள் அப்படின்னு சொல்லுவோம். மீனோட செதில்கள். We will remove all these scales before cooking, right? So those scales will play a vital role in defining its morphology or identifying the fish species, and also various functions will be performed by scales. We can even identify the age of the fish by examining the scales. Okay, so these scales are basically three types of scales are found in all the fishes. Number one is placoid scales, which is found in sharks and rays. Those placoid scales are found in elasmobranchs, that is sharks and rays. And number two is ganoid scales. Ganoid scales is found in garfish, which is a primitive fish. And the other one is elasmoid scales, which is found in the bony fishes, which is teleost fishes. These elasmoid scales are further divided into cycloid and ctenoid. This ctenoid variant of scales are found in perches. Perches, for example, sangaramin, sangaramin. Then also sea bass, Latus calcarifer. இதுல எல்லாம் பார்த்தோம்னா ctenoid scales இருக்கும். Whereas cycloid scales are present in the rest of the bony fish species. கிட்டத்தட்ட அநேக மீன்களுக்கு இந்த cycloid fishes தான் இருக்கும். Cycloid scales தான் இருக்கும். ஓகே, ஸோ அதெல்லாம் மார்ஃபலாஜிக்கலி வெவ்வேறு விதமாக இருக்கும் பார்க்கறதுக்கும் சரி. அதை அனலைஸ் பண்ணி நம்ம பார்த்தோம் அப்படின்னா ஒரு ஃபிஷ்ஷும் வெவ்வேறு ஷேப்பில் வெவ்வேறு ஃபீச்சர்ஸோட இருக்கும். அண்ட் அதை வச்சு தான் இந்த மாதிரி 3 பேசிக் டைப்பாக டிவைட் பண்ணியிருக்கோம்: placoid, ganoid and elasmoid. The third type, elasmoid, is further divided into 2 types, which is cycloid and ctenoid. That's it. We have seen almost the most important morphological features of a typical fish. Thank you, one and all. Thanks for listening.